Chapter 2 Biological Classification. Since the dawn of civilization, there have been many attempts to classify living organisms. It was done instinctively, not using criteria that were scientific but born out of a need to use organisms for our own use, for food, shelter, and clothing. Aristotle was the earliest to attempt a more scientific basis of classification. He used simple morphological characters to classify plants into trees, shrubs, and herbs. He also divided animals into two groups, those which had red blood and those that did not. In Linnaeus' time, a two kingdoms system of classification with Plantae and Animalia kingdoms was developed that included all plants and animals respectively. This system did not distinguish between the eukaryotes and the prokaryotes, unicellular and multicellular organisms, and photosynthetic green algae and non photosynthetic fungi organisms. Classification of organisms into plants and animals was easily done and was easy to understand, but a large number of organisms did not fall into either category. Hence, the two kingdom classification was used for a long time was found inadequate. Besides gross morphology, a need was felt a need was also felt for including other characters like cell structure, nature of wall, mode of nutrition, habitat, methods of reproduction, evolutionary relationships, etc. Classification system for the living organisms have hence undergone several changes over time. Though plant and animal kingdoms have been a constant under all different systems, the understanding of what groups organisms be included under these kingdoms have been changing. The number and nature of other kingdoms have also been understood differently by different scientists over time. Table 2.1 Characteristics of five kingdoms. Five kingdoms: Monera, Protista, Fungi, Plantae, Animalia. Cell type: prokaryotic in Monera, eukaryotic in all other kingdoms. Cell wall in Monera is non-cellulosic, that is polysaccharide plus amino acids. In Protista, it is present in some. Fungi, cell wall of chitin is present. Plantae, cellulosic cell wall is present. Animalia, cell wall is absent. Monera, cell membrane is absent. In all others, cell membrane is present. That is, all are eukaryotes. Body organization in Monera is cellular. In Protista also it's cellular. In fungi it is multicellular or loose tissue. Plantae it is tissue or organ. In Animalia it is tissue, organ or organ system. Mode of nutrition. In Monera, autotrophic, chemosynthetic and photosynthetic, and heterotrophic, saprophytic and parasitic is present. In Protista, autotrophic, photosynthetic and heterotrophic is present. Fungi, heterotrophic, that is saprophytic or parasitic is present. In Plantae, autotrophic, photosynthetic is present. In Animalia, heterotrophic, holozoic, saprophytic, etc. are present. R.H. Whittaker in 1969 proposed a five-kingdom classification. The kingdoms defined by him were named Monera, Protista, Fungi, Plantae, and Animalia. The main criteria for classification used by him include cell structure, body organization, mode of nutrition, reproduction, and phylogenetic relationships. The three-domain system has also been proposed that divides the kingdom Monera into two domains, leaving the remaining eukaryotic kingdoms in the third domain and thereby a six kingdom classification. You will learn about this system in detail in higher classes. Let us look at the five kingdom classification to understand the issues and considerations that influence the classification earlier. Earlier, classification systems included bacteria, blue-green algae, fungi, mosses, ferns, gymnosperms, and the angiosperms under plants. The character that unified the whole kingdom was that all organisms included had a cell wall in their cells. This placed together groups which widely differed in other characteristics. It brought together the prokaryotic bacteria and the blue green algae cyanobacteria with other groups which were eukaryotic. It also grouped together the unicellular organisms and the multicellular ones. Say, for example, Chlamydomonas and Spirogyra were placed together under algae. The classification did not differentiate between the heterotrophic group fungi and the autotrophic green plants, though they also showed a characteristic difference in their cell walls composition. The fungi had chitin in their walls, while the green plants had a cellulosic cell wall. When such characteristics were considered, the fungi were placed in a separate kingdom, kingdom fungi. All prokaryotic organisms were grouped together under kingdom Monera, and the unicellular eukaryotic organisms were placed in kingdom Protista. Kingdom Protista had brought together Chlamydomonas, chlorella, earlier placed in algae, within plants and both having a cell wall, with Paramecium and amoeba, which were earlier placed in animal kingdom, which lack cell wall. It has put together organisms which in earlier classifications were placed in different kingdoms. This happened because the criteria of our classification changed. This kind of changes will take place in future too, depending on the improvement in our understanding of characteristics and evolutionary relationships. Over time, an attempt has been made to evolve a classification system that reflects not only the morphological, physiological, and reproductive similarities but is also phylogenetic, that is based on evolutionary relationships. In this chapter, we will study characteristics of kingdom Monera, Protista, and fungi of the Whittaker classification, Whittaker system of classification. The kingdoms Plantae and Animalia, commonly referred to as plant and animal kingdoms respectively, will be dealt